Vitex segundo, or locally known as lagundi, is very popularly studied in the Philippines. In fact, it is being processed as teas, tablets, and capsules to treat asthma and other respiratory-related illness. Previous researches reported that agnoside is a main component of lagundi, and recently the Department of Science and Technology reported that lagundi showed positive results in the treatment of COVID-19. These important developments call for the development of a reliable method that will analyze lagundi and its medicinal preparations. This inspired me to pursue my research entitled Development and Validation of an Hplc-Dad Method for the Chemical Fingerprinting and Quantification of Agnoside in Vitex segundo lagundi leaves, and commercial tablets. And I am Michael Casas, the Chief of the Student Services Division of Philippine Science High School, Southern Mindanao Campus. So why is this study important and worthy of focus? Number one, Hplc-Dad is sensitive and a reliable method for the analysis of lagundi and its medicinal preparations. Second, this will allow for a stronger enforcement of quality control standards that will propel the development of lagundi and other medicinal plants in the traditional medicine area. And lastly, this will allow for the updating of the Philippine Pharmacopoeia to incorporate more advanced methods for the analysis of lagundi and the other medicinal plants. To accomplish the research, we have set three main objectives. First, we would like to develop an Hplc-Dad method that could quantify agnoside and prepare a chemical fingerprint from lagundi and its medicinal preparations. Second, we would like to validate the developed method to conform to set international standards. And lastly, we would like to apply the developed and validated method to real samples to check its applicability. The research objectives were accomplished using the following schemes. For the method development, for the method validation, and for the method application. The following results were found from the research. For the method development section of the study, we have developed a gradient elution Hplc-Dad method that allows for the separation of the components of lagundi and allowed for the elution of the most dominant peak, which was later found to be agnoside at 14 minutes. Second, for the method validation section of the study, the method was found to be compliant to ICH and chemical fingerprinting standards, allowing us to have the confidence that the method can be used reliably for the analysis of lagundi and its medicinal preparations. Finally, in the method application section of the study, it was found that reflux using alcohol and decoction, which is boiling the samples in water, allows for the maximum extraction of agnoside from the sample. Moreover, the chemical fingerprint prepared for lagundi and its therapeutic preparations allowed for the differentiation and easy identification of lagundi and differentiation from all other plant samples. In conclusion, a gradient elution method was developed using Hplc-Dad for the chemical fingerprinting and quantification of agnoside in lagundi and its medicinal preparations. Moreover, the prepared method was validated using ICH and chemical fingerprinting standards and was found to be compliant. Finally, the research also applied the method to real lagundi samples and successfully quantified agnoside and prepared the chemical fingerprint of the samples for easy identification and differentiation. Overall, the research recommends to explore other analytical methods to analyze lagundi and its medicinal preparations. Moreover, the analysis of other medicinal plants should be underway. Daghang salamat and mabuhay ang Pilipinas!